So welcome to another Bioinformatics live session. So today is a relatively easy one, just to sort of ease us into the Christmas break, and it's just about downloading sequencing data, and primarily from the Sequence Read Archive, the SRA. And just briefly, so we are going to continue these sessions into the new year, but we're going to take a little break in January. The February session will then be all about SnakeMake, which is a sort of derivative of Python, which is used for building bioinformatics pipelines, and that'll be presented by Dan. And then we don't really have a plan from there on in, but we've been discussing doing a few more interactive things, such as drop-in sessions where people can just come with their own data or problems, and we'll just be there to help out, or some sort of data analysis session where we might set a challenge and see how people approach that differently. We might even have some social sessions, so coffee mornings or pub lunches, just to try and get a bit more networking and socializing between people that are performing bioinformatics and doing bioinformatics in the center. And as always, we're always looking for future topics. So we have sort of an ideas board. We are looking to do things on Git, which is version control for your scripts, Nextflow pipelines. Hopefully going to get someone in to demonstrate AlphaFold, as well as using the Eddy, the cluster ECDF. But if you have any ideas of your own or anything you would like to present or for us to cover in these sessions, then please get in touch. So as I said, the topic for today is just downloading sequencing data from the SRA, which is the Sequence Read Archive hosted by NCBI. And so we'll be having a look at the website and exploring that a little bit. We'll also be looking at GEO, which is the Gene Expression Omnibus. Essentially, most people will probably go through GEO to find sequencing data in the SRA. And then we'll be looking at some tools for actually downloading sequences. So the SRA tools are produced by NCBI themselves. So that's a set of command line tools for downloading sequencing data. There is also this quite useful application called the SRA Explorer, which we can use. It's a third-party website, but it searches through SRA and alternative websites and gives you links for downloading data. And I'll also just sort of touch on what the European Nucleotide Archive is and how that's related to SRA. So first of all, the Sequence Read Archive is a database or a repository for all sequencing data. It hosts all types of data, so long reads, short reads from all types of sequencing platforms, paired end data, single end data, etc. And all of that raw data is organized and given unique identifiers. So an SRA identifier starts with SR and then followed by one of these letters. So there are projects or studies, which are sort of collections of data, which will contain one or more different samples. Samples are labeled as SRS, and they could relate to one or more experiments. And then an experiment hosts the metadata and the sequencing runs for sort of one sample. And this could contain one or more runs. In that case, these runs would be technical replicates. And the SRR relates to a run itself. And so this is where the actual data is. So you kind of see in this little screenshot here that this SRR number relates to a single run, and then this .1 is the first read, and we can see the read sequence here. So we can view all that through the SRA. This data is stored in the SRA in the SRA format, which is a really sort of highly compressed format, which makes it really easy to view on their website. So it's not natively stored as FASTQ files, for instance. Instead, the reads are saved as what they call spots, and a spot may contain a single read or a paired end read. but we'll look at that a little bit later. So I mentioned GEO as well. So that's a gene expression omnibus, and this is a genomics data repository for high throughput experiments. So it sort of started with microarrays and is now hosting all types of data from high throughput sequencing experiments. And so a GEO record typically has the experimental metadata, some form of process data, which is, often the data that is used to create images and tables and publications. It will also have links to the raw data sets. So if that sequencing data will be stored in the SRA, and links to any publication if the data has been published. And in GEO, the data is organized into series or super series which contain multiple series. And within each series, you find samples, which then relate to an individual sequencing sample. And so often when we publish data, we submit our raw data and our process data to GEO. And GEO actually handle the processing of raw data and create records in the SRA, the sequence read archive, for your raw sequence files. And so what you will find in most publications in often a data availability section is a GEO ID, which will link to a GEO series. And each of those GEO series will link to an SRA record for a project. And each GEO sample will be linked to an SRA run or experiment. What I should say is that you can also go into the NCBI databases and search those directly for data sets. So NCBI has this advanced search builder where it uses a specific syntax for searching different fields that are in all their different databases. So you can search by author, by organism, by experiment type, for instance. And they have the same sort of syntax and search available for SRA, GEO, PubMed, DBSNP, so all these databases. And that searching strategy can be used on their website, but also programmatically. So if you are using APIs or R or Python to query the NCBI, then learning how to build those queries can be quite useful for you. But there is documentation on how to do that, and we won't go too much into that today. So then as far as downloading data from the SRA, you can, in some ways, directly download data through the website. There are lots of links for sending data from the SRA directly to cloud platforms such as Amazon Web Services, or you can download data via the browser, but it's very limited. So it's limited to 5 gigabases. So that's the number of DNA bases rather than gigabytes. And so not most data sets you won't be able to download via the web browser itself. The SRA has a tool called the run selector. Again, this allows you to sort of select many different data sets at the same time. But again, you can only send these to cloud platforms or download the metadata or the accession IDs, which might help you later on. There is this third-party tool called SRA Explorer, which we'll look at. This, again, you can search for data sets in SRA and get the metadata, but it also gives you direct URLs to where the FASTQ files are stored. So you can use these to sort of FTP services or some sort of HTTP download to get these FASTQ files. And lastly, which is the method that we use mostly, is the SRA tool. So this is a command line application which installs on Linux. It allows you to download files directly from the SRA so in SRA format or FASTQ file format by using the tools FASTQ dump, prefetch, or FASTQ dump. So FASTQ dump was the sort of original version, and this would directly download FASTQ files from the SRA website. The newer version uses prefetch, which downloads the SRA file, which, as I mentioned, is highly compressed, so it's a lot smaller. And then FastQ will convert that into FastQ format on your machine. So it's a lot quicker and it relies less on their network connection. So where FastQ can fail if a download is taking a long time, prefetch will download the data really quickly, and then you can convert it on your own local machine using FastQ. And so we will look at using these today as well. And the last thing that I really want to mention is that all of the data in SRA is hosted by NCBI, which is part of this International Nucleotide Sequence Database collaboration. And so this is a joint collaboration between NCBI, the European Bioinformatics Institute, and the DNA Database Japan. So all three of these organizations host data which is shared with all three databases. So their EBI have equivalents of SRA and GEO, but all of the data that exists in them also exists through the NCBI website. So at EBI, we have the European Nucleotide Archive, which is analogous to SRA. It hosts sequencing data. Any data that is submitted directly to ENA then gets an ENA ID instead of an SRA ID. But you can search the ENA database to also find SRA records and links because the data is shared. The sort of geo equivalent is called Array Express. So that's for high throughput genomics data. And the EBI also have this biostudies database, which is actually quite useful. So what they're trying to do there is create one record for all the different data types that are in a study. So obviously a lot of projects will not just be sequencing data, there might be imaging data or electron microscopy data as well. So the biostudies record tries to collect records from all these different submissions to different types of data repositories. Okay. So we are going to go ahead and just look at the SRA database. I'm actually going to start by looking at a paper. I can go right to the top. Okay, so this is a paper that Jose is probably familiar with. So this was published by Eric Shermer's lab. I'm on it. Jose's on it as well. Rafael was the first author. And so in this paper, there was RNA-seq analysis of wild type and knockout mice with this gene TMEM120A knocked out. It's known that this protein knockout disrupts fat genome organization in adipocytes. And so there was differential expression analysis performed on wild type and knockout mice fed on two different diets, low fat and high fat. And so generally, when we want to find sequencing data, it's probably related to a publication that we're interested in. And so most publications will have some sort of data availability section. So if we click on that, then we can find often geo links, okay? So if you want to find where the data is, just a quick command find on data availability, or just type the word geo and you'll find a section very similar to this. So I can see that the RNA-seq data sets are available through geo at this identifier. So we're going to go ahead and click that. And this takes us to the geo series record. So we can see this begins with the GSE. We get a little bit of information about the experiment, an abstract, a link to the publication, and some of the metadata. We then have the samples. So these are GSM. So these are the individual samples. We can see the wild type, the knockout, and the two different diets here. If we scroll down a little bit, we can see a link to the SRA. So this is an SRP ID, which means it is the study or the project entry. And we also have some process data from this experiment as well. So this is the salmon TPM file. So that will give us all the gene expression levels for all of our data sets at once. If we click on an individual sample, so I'll just go into the first one here. So we're now looking at one sample. It gives us some information on all the data processing that went in to process that sample. And again, we get an SRA link to an experiment. So this will be for the individual experiment, and also some process data, which we can download directly from Geo. These are bigwig files for that data set on the forward and reverse strand. So let's just have a look at the SRA entry here for the experiment. And we'll also go to this tab and look at the SRA entry for the study. Okay, so this SRA identifier shows us all of the SRA entries which relate to this project. And so again, we can see that we have the 20 samples there. Let's just go into that one that we created. So this is the experiment entry. We can see again that there is a geo identifier that we could always link to go back to Geo. We have a little bit of information about the study, and we can see the study ID. We could ask for all experiments or all sequencing runs that belong to that study or project. Similarly for the sample, a little bit of information on just on the platform and strategy that was used to produce this data set, the geo accession, and then we have a run. So in this case, there's only one run. If we had multiple runs, then it might represent different lanes on a sequencing machine. So these might be sort of technical replicates. We can see that there are 160 million spots. So it's 160 million reads. And it equates to around 50 gigabases and around 14 gigs of data. So we'll click on the run ID. And let it load. Okay. So again, we just get some information about the data size and the read itself. We can see a bit more about the experiment. So it's RNA-seq, cDNA, and we can see that it's paired end sequencing as well. Further links to the sample information and links to the bioproject study as well. So that's similar to the biostudies project from EBI. There's a few tabs here. So analysis, this is just basic analysis that runs every time an SRA entry is created. It's probably not all that useful. It's just sort of mapping against different things. We can see it's just sort of a default analysis, and there isn't much to it. You can go into this tab and actually load the individual reads. I'm not sure exactly why this is useful. We have 16 million pages of reads because there's 160 million of them, but we can, see read 1, read 2, and we can see that these are paired end, for instance. There's a data access tab, but this only shows you sort of links to cloud platforms, as I mentioned, so we can directly see this sequencing run on AWS, the Amazon Web Systems. There is a FastA FastQ download, but again, what we'll see is because this is large, it exceeds the web browser download limit of 5 gigabases and tells us to use the SRA toolkit to download and to do that locally on our machine. Okay, so we've looked through sequence read archive, we've found an individual run, that's great, but at this point, we don't really know how to download it. And so we're going to try and use some other tools to do that. Firstly, though, what I want to show you is just the advanced search in if we go to the SRA home, first of all, we start with the search bar. So you may want to query SRA directly for data sets or geo, if it's the case that you're trying to find some data that relates to your experiment or that you would like to analyze and you haven't found it through a publication. It's always useful just to go into this advanced search tab. And the NCBI query builder then allows you to search by all different fields. So for instance, if we select author, I can search for, so I'll try to find a different geo series that I know exists. So I'll search for one in the bird lab. We are interested in mouse data, so I can just put Muzz in there under organism, and strategy will look for RNA-seq, and the bird lab work on MECP2. So I'm just going to put that in there, and you'll see that it's building up this sort of query structure, which is standardized across all the NCBI databases. And I'll hit search on that. Oh, maybe I had to do RNA-seq. Okay. So from that, the first hit here is the sequencing run. So I'm going to click on that. It gives us a geo accession ID. So let's look at the geo page as well. Go back to geo. Okay, so we can see here we have an individual sample. If we scroll down, we can see that which series these belong to. So I'm just going to click on this series here. And now I have a list of all of the samples that were part of that study. So I can see the knockout and wild types. We can also see that this is a super series and it actually contains two subseries. So there is attack-seq data and RNA-seq data. So if I wanted to look at the attack-seq data only, I could go into this geo series, and this gives me all of the samples from the attack-seq. Okay. And again, there will be SRA links for all of this data as well. So yeah, so just sort of useful ways to search through NCBI databases directly to find the sequencing data that you want. The alternative is this tool here called the SRA Explorer. Okay, and I'll just refresh. So SRA Explorer is written by this bioinformatician who's quite sort of famous in the field for producing things like the NF-core pipelines, and also worked on things like FastQC, etc. It's a really sort of simple website just for searching through SRA. It works a little bit like a store where we can search by an accession number, or use search terms like we did in NCBI. So for instance, if I did the search again, bird, author, Muzz, organism, and then I'll just put RNA, seq, and MECP2. If I hit search, okay, so we can see that it sort of translates automatically my search into something very similar to what we had in the NCBI query builder, and it finds 82 results. If I, yeah, so it's finding, yeah, so actually if I come down here, this is the ones that we were looking for here. So there must be several other MECP2 RNA-seq related data sets with an author called Bird, but we can see this is the geo entry that we were looking for. And again, we get links to the SRA run. Other ways you can search, so you could put in a geo accession. Okay, and again, we're getting this data here. We could use a geo series ID to get multiple data sets. We could use any SRA project or experimental or run IDs, or we could just do a basic text search as well. So let's go back to this run that we looked at in SRA and have a look in SRA Explorer, see what comes up. Okay, so what we can do is we can click this button here and we have this add to collection. And we'll see that I now have one saved data set in my cart. So that way I could search and find multiple data sets and keep adding them to my cart here. Actually, maybe we should add a few more. We'll just go to the series, RNA-seq series, and get that ID. Sorry, I don't know why this isn't loading. Well, we'll try a different method. Try this project. Okay, there we go. So we can select multiple data sets, add them to our collection, and then go to our cart. And so here we have three tabs. One, we can download the metadata. We can also download the SRA files themselves, so raw SRA format, or the FASTQs, which is probably in most cases what we're interested in. And so this actually goes through the ENA database, which makes the SRA FASTQ files accessible and produces a list of URLs in this case, which we can then copy directly to the FASTQ files. So we can see there is a 1 and a 2 for each sample. It can also give you a bash script, which you can use, which will run curl to fetch these directly off the internet, download them from the FTP side and rename them appropriately. A few other links for things like SRA, which is a sort of FTP download program, ClusterFlow, and the BC Bio, which are other bioinformatics applications. So in this way, you can fetch URLs for directly downloading the FASTQ files. So that's great, but as I did mention, again, you have an issue of directly downloading large FASTQ files off the internet. And so it can take a while and there can be connection errors, which means that your download could stop or run into issues at any point. And so the sort of recommended approach, if you have access to a Linux command line or to our servers, is actually to use the SRA tools. And so these are produced by NCBI, and it's the SRA toolkit. So I'm just going to stop sharing and share my terminal. Okay, so I am currently logged into BIFX Core 2. Okay, and I am going to download this series, this sequencing run here. So I should show you this first. So we'll go to SRA and we'll just search for this run ID. The reason I've chosen this one is because it is quite small. So we can see there's 10.4 megabases. It's only 5.2 megabases of data. So we should be able to download that pretty quickly. So yeah, 5.2 megabases. So in my terminal, as I mentioned, there are a couple of tools. So there is FastQ dump, and there is the newer version, which is faster QDump. Okay, so for both of these tools, all we really need to do is write fast QDump or faster QDump. Make this a little bit bigger. And then the accession number, and this will start downloading directly from the SRA in FastQ format. So I'm just going to remove, because I've already downloaded it. So I'm just going to remove these. Okay. So I can start doing this, but it will take a couple of minutes. So faster QDump will directly download. So what it's doing is fetching the SRA file, converting it to FastQ, and then downloading the FastQ. So the alternative is to use the newer tool, which is called prefetch, and to give the ID. Okay, and what we can see, so this is downloading right now, and that's it done. So if I just do an ls, I can see that I now have this folder SRR with the ID. So let's have a look at what's in there. And we have this SRA file, which is 5.3 megabytes of data, which is similar to what we saw. So that downloaded very quickly. So that's me prefetching the data. And now when I run faster QDump on the same ID, it will look for this folder that exists, and it will go in and it will convert this SRA file to FastQ format. And so that did that really, really quickly. And now when I have a look at my directory, I now have, for this ID, I have an _1 because it's a paired end data, _1 and _2 file. We can see these are both 12 megabases each, so around 24 megabytes of data, sorry, which is a lot larger than the 5.3 that we had to download to begin with. And this is a very small data set. So you can imagine that once you get up to data sets that have 100 million reads, that the sizes scale, quite hugely between the FastQ files and the SRA files. And so the best thing is to prefetch and then run faster QDump. By default, faster QDump is set to use the split 3 option. And what that means is it looks at the spots in the SRA file and looks to separate any reads into different files. So you'll get an _1 for the first pair, an _2 for the second pair. You will also get a third file if any reads exist in there that don't have a mate or a pair. So what you will find sometimes is that people upload data to SRA, which maybe isn't strictly the raw data. It may have been pre-filtered at some point. And so you may get files that have paired reads, but also single reads as well. And so that can cause sort of downstream applications to fail when you're processing the data. So the default in FastQDump is to use the split 3, which will split them by read, and also the singletons in their own file. That wasn't the default for FastQDump. We'll just have a little look at the help text for FastQDump. And so we can see that we can set things like the out file if we want it to have a different name. We can also set a directory where we want to save the FastQ files. There are these 3 options here. So split spot will split your spots into reads, but it will write them into the same file. So bizarrely, the spots are kind of saved as concatenated text. And so this was the default in FastQDump was just to write out a whole spot, which didn't really make much sense. So you had to set one of these options. So this would essentially give you like an interleaved paired end file if you had paired end data. The split files will write into 2 files, so _1, _2. And the split 3 will also write these single reads into its own special file. So this one probably makes the most sense, and it is the default now for FastQDump. So yeah, that makes it all quite useful and easy to use. Okay. So that's just a quick example of running FastQDump and prefetch to get your data on a single run. What I tend to do if you want to look at multiple sets is to sort of find the data in something like SRA Explorer, where you can then download multiple accession IDs into a text file. You can also use, so you'll find on Geo a link to the SRA run selector, which is kind of similar to SRA Explorer, I guess. Again, they have so you can see all of the data sets, or you can select a few that you want, and it will come up here and selected. You can download the metadata. You can download the accession IDs here. You can deliver data to cloud platforms as well. But for instance, you can select the ones you want. You can click on accession list, and that will download just a text file of all the SRR IDs. And then you could write a little script which will run prefetch and FastQDump on each of those IDs to download the data either sequentially or in parallel. Yeah, the one other thing to say is that by default, FastQDump, well, it only writes your data into a text format, so as a FastQ file. FastQDump had a --gzip option, which would compress it on the fly, but again, this just meant that the whole process of downloading and writing took a lot longer. So generally, after downloading your FastQ files, the last thing you want to do is then probably gzip them all into a compressed format. Okay, so that is pretty much everything I wanted to cover today. So what is SRA? What is Geo? How do we find data? How do we navigate those databases? How do we get lists of sequencing runs that we might want to download? And then using the SRA toolkit to download the data. Does anyone have any questions on that at all? Nope. Okay, great. In that case, so as always, I will stick around for another until 3 o'clock if need be. If anyone has any questions about anything unrelated or their own bioinformatics analysis, then feel free to ask. Otherwise, I would say have a Merry Christmas, and we will be back again with these sessions in February. Thank you very much.